Good morning, everybody, and hope you all had a good Diwali. So, in this class, I'll try to wrap up the MLP topic or deep learning topic. So, we have seen that you can train deep networks on very hard classification problems. And what is interesting is people found that since deep networks show comparable performance to humans, right, they become a very good potential candidates as models of sensory function in the brain. So there's a nice review by Kell and McDermott. So it's called Deep Neural Network Models of Sensory Systems. Windows onto the role of task constraints. So the thing is, if you train deep networks or deep neural networks on certain classification problems, which represent standard sensory processing in the brain, and if you constrain those models, the training in certain fashion, then the way the networks learn the problem, right, emulates some properties of brain function. So there's a lot of parallels between what the networks learn and how they learn, and how the real brains represent the information when they process that sensory input. So so in this review, the authors look at several such studies. It's a review paper. They look at many such published literature studies and show these parallels between DNNs and sensory function. They say that this is possible because it's become possible because of superior computing power that we have with GPUs and all that. And also they start with the idea that CNNs trained on sort of this sensory processing tasks or classification tasks have achieved human level performance. That is the starting point of this whole comparison. So this is just a standard picture of a CNN. So you know that CNN has initially a bunch of convolution layers followed by max pool layers. So you have convolution and then some kind of nonlinearity, either rectification or a ReLU or a sigmoid or something like that, and then a max pooling, then again convolution. So you have these layers occurring alternatively, and then you have few such stages like that, and after that you have fully connected layers and then a softmax layer for classification. So what they have done in this particular plot, the results they're showing is, so they have a bunch of images that need to be classified. There's a huge data set of images where for the same object, they have many images taken so that there is variation in lighting, variation in background and things like that. So there are two types of classification problem. One is to classify the image itself, right? And the other is to classify the object that is contained in the image. So you only have to classify the object of the image. So which means object level classification is actually at a higher level. It's a broader classification. Image level classification is more fine grained. So they train various kinds of network models on both these classification problems based on the same data set. And they look at consistency, compare with consistency, and they compare with humans. That is, so you give the same data set to humans and ask them to solve the problem, to classify it. So you initially specify whether the classification has to be done at object level or at image level. Then humans also make some mistakes. So look at that error pattern made by humans and look at the error pattern made by the network and perform some kind of a correlation between these two. Right? Then that will give an idea of how far is the network's error making pattern similar to the human's error making pattern or error pattern. So that is called consistency with humans. So what they found is when you look at the classification done at image level, the error pattern between humans and networks is not very close. That the error signature is very different. So which is seen in the right plot here. But when you compare it at object level, there's a lot of correlation between networks and humans, and that correlation is seen for a wide variety of deep networks: LXNet, NYU, VGG, GoogleNet, and so on and so forth. So what this teaches us is, it doesn't matter what the exact architecture is, as long as architecture has some kind of a hierarchical layered structure and trained on the right kind of a problem. So this is where the task constraint comes into picture. So when they trained it on at a single at image level, then it was very bad. But when they trained it on object level, it was very similar. The performance pattern, performance error signature between humans and networks is very similar. And you see that all these networks give about the same kind of consistency with humans. Another thing is among humans themselves, there is no perfect consistency. It's not that all humans made exactly the same kind of had same kind of error signature. So that is shown in this dashed gray dashed line, which is a horizontal line. So it's not one, so which means all humans didn't make the same kind of error. Okay, so what is amazing is the network consistency with respect to humans and the within human consistency is about the same, which means the networks are behaving very much like humans as far as the performance is concerned. So we're not talking about what kind of representations the networks have generated. So for example, when humans solve say image classification problem, so we know from the last few classes that there is the retinal image which goes to LGN and then to V1 and goes via a series of cortical layers and finally it terminates in inferotemporal cortex, and that's where the complex objects are classified. But then there is no V1, there is no V2, there is no IT area in a convolutional network. So how can you even compare? So that question will come a bit later, right? But what is interesting is it's enough if the network has a sufficiently deep hierarchy, right? And then if it's enough if you constrain the problem in certain fashion, then at least at performance level, both network and humans behave similarly. Okay, so that is a very good first step. So now let us look at go deeper into the network and see if there's any connection at all between what the network hidden layers are representing, right, and what is happening in the brain. Um, okay, so look at the left plot. So the left plot is about so you take activity recordings from B4 part of macaque is a kind of a monkey, it's a primate. So you take recordings from B4 and it is also trained on the same kind of a kind of image set of images, right? And the network is also trained on the same set of images. Now what we are asking is, does layer one have enough information? So they take the outputs of, so you present an image to the network, present the same image to the monkey, to the macaque. Now in the case of macaque, take the recordings of neural activity from V4 area. In case of network, take the outputs of the neurons from different layers. So layer 1, layer 2, layer 3, and layer 4, and so on. So now the question is, given the activity of any layer, let's say I'm taking layer 1, given the activity of layer 1, from this I'll just construct a linear transformation, right, and try to predict the activities observed in V4. How good can I get, and how good will that fit be? So that is what is marked. Some kind of an error is shown here. So from layer 1 of the convolutional network, if we try to predict the activities of V4, V4 is considered slightly a higher level in the visual hierarchy. So layer 1 in convolutional network is at lower level. So it's interesting that although the layers of the convolutional network have nothing to do with the biological layers of the visual system, but the hierarchy is very similar. That's what it looks like. So layer 1 neurons are not able to predict the activities of V4 neurons. But layer 2 neurons, right, in the convolutional network are very well able to predict the activities of V4, and layer 3 is even better. Layer 4 again is not very good because the prediction levels have gone down. So similarly, if you look at the predictions of IT is where we have we have seen that you have neurons that respond to complex objects, and in layer 4 in a convolutional network we are already pretty close to the output layer. So most of the neurons there have sufficiently large receptive fields that they can look at the entire input image and can represent the whole class of the object. So again, that intuitive expectation is also quite justified and quite validated by the results. So layer 1 gives very poor prediction, and then next layer 2, layer 3, layer 4. So you can see that there is clearly a hierarchy within the neural network, and which matches very well with the hierarchy of the visual system. So So layer 4, is very similar to the IT area. So they're not comparing the actual activity levels of layer 4 and IT area, because that won't make any sense, because layer 4 is, the convolutional network is not designed to look like the brain. But they're looking at the ability to predict one with the help of the other using a simple linear transformation. So basically you're trying to multiply the activities of layer 4 neurons with a matrix and then try to estimate IT activities. And that comes out to be pretty good. So then that is from electrophysiology, from probably the electrical recordings of neurons. A similar thing done in case of humans with fMRI, functional magnetic resonance imaging activity. So basically fMRI is based on the idea that when neurons are active in the brain, they result in local increase in blood flow, because for neural function you need energy. Energy comes from blood glucose. So increased neural activity leads to local increase in blood flow. So that's what fMRI is based on. So they look at different areas of visual cortex in their activity levels as seen from fMRI, and then compare that with right, again do the same exercise with a deep network. Again they get very similar results. Okay, so next they also got similar results in case of auditory system, which is even more surprising, because so people have used CNNs to perform uh, visual classification tasks forever and ever. I mean, since the early days when the CNN was first proposed in the late 90s, right? It was has been used for image pattern recognition. But auditory is somewhat rare, and it's quite surprising that they can even explain auditory function in real brain. Okay, now so we know that sound is created by pressure waves in air, and they have frequency, amplitude, and all these properties. This is only high school, so I don't spend time on this. So sounds are processed starting with the ear, right? And so it is the ear machinery that converts these pressure waves called sounds into electrical signals, which are then processed by the brain. So here you have again, if you recall your high school biology, there's outer ear, middle ear, and the inner ear. In the outer ear, the basically air, the waves propagate to this kind of a channel, right? And the auditory canal. At the end of it, you have the tympanum, or what is popularly called the eardrum. So the eardrum vibrates to the pressure waves, and behind that you have this set of three bones called the ossicles, right? Malleus, incus, and stapes. So the last bone, which is stapes, which looks like a ring, right? Like you have this some kind of a ringer on a door with which you can bang on the door. So it looks something like that. Even its function is something like that. So you can see this picture better in the figure on the right side. So this is stapes, this bone stapes, which is attached to another inner wall. And inside this you have a special structure called basilar membrane. So let us look at that. So this is cochlea. So this whole thing is cochlea. The cochlea is actually looks like a snail. It's all curled up with lots of coils. But for simplicity, for explanation, you just imagine it's all extended and stretched out. So the stapes, when it kind of vibrates in sync with the tympanum, right? It also introduces those vibrations into this fluid medium which surrounds the basilar membrane. This medium is called endolymph. Now the physics of the basilar membrane is such that depending upon the frequency of this oscillation or vibration of stapes, a specific part of the basilar membrane also vibrates. Okay, so that will look something like this as shown in this figure here. High frequencies produce vibrations close to the mouth of this structure, and low frequencies produce vibration far at the far end. Now there are these cells called hair cells, right, which are attached to the basal membrane on one side and to the kind of roof of this whole structure on the other side. So whenever the membrane vibrates, these hair cells are stretched, and that stretch causes change in electrical potential inside these cells. Because I don't know if I mentioned that there are certain kinds of ion channels. So you know about sodium potassium channels and all that. There are certain kinds of ion channels which open when the membrane is stretched. It's like, ion channel is like a little hole in the membrane. When you stretch the membrane, that hole gets widened. So that allows a passage of ions. So these stretch-sensitive ion channels work like that. So there are such channels in these hair cells. So therefore, this mechanical changes in stress uh, get converted into electrical signals, and that's what is propagated via the auditory nerve, to higher brain areas. So you can see all these auditory nerves originating from the cochlea and sending outputs to the next stage. So even at this level, you can see since the cochlea or the basilar membrane is tuned to different frequencies. But you can see that suppose I introduce a sound which has two frequency components, F1 and F2. That will produce two local vibrations, right, in this membrane. So it look like it's performing some kind of a Fourier transform of your input sound. Okay, so it's a very nice mechanical linear structure which is effectively doing some kind of Fourier transform. So therefore, that kind of transform signals are now propagated upwards, right towards the cortex. These signals go through again a hierarchy. The very first stopover are called the cochlear nuclei. There are two of them: dorsal cochlear nuclei and ventral cochlear nuclei, DCN and VCN. So from there, some fibers directly go to this stage here, the inferior colliculus, which is next to another important structure called the superior colliculus. Superior colliculus is actually closely connected to the visual system. It controls eye movements. Inferior colliculus is connected to the auditory system. Right there, it takes signals coming from the cochlear nuclei and sends them further on into the brain proper. So on the way, some parallel fibers from VCN and DCN also go to the superior olivary nuclei, which is right here. And so again, from superior olivary nuclei, you again project to the. So this is like a little detour. Project inferior colliculus. Inferior colliculus inputs now enter the brain proper, and as you know, most sensory input going into the brain has to go through this big junction box called the thalamus. And as you remember, in the case of visual input, that the nucleus of thalamus is a lateral geniculate nucleus. In case of auditory system, that nucleus is medial geniculate nucleus or medial geniculate body. So from MGN or MGB, right, information now goes finally to the auditory cortex or primary auditory cortex. Okay, so you can see that here also there's a kind of a hierarchy. So this is just same description. This picture shows the hierarchy starting from cochlea, the cochlear nuclei, DCN and VCN, and then superior, inferior colliculus. Then you have the medial geniculate body or MGB in thalamus, and finally your auditory cortex. And beyond that, all the sensory inputs, so they're most abstract representations, right, refer to the prefrontal cortex. Okay, so yeah, so in addition to just classifying sounds, and you can also localize sounds, and so that is where is the sound coming from. So some of that function can occurs inside the superior olive because it responds to uh, time difference in the arrival of sounds. Because how do you find out where the sound is coming from? Suppose sound is coming from your right. Right, the sound reaches your right ear first, right, and then after a delay it reaches the left ear. So that's a cue that it's coming from the right ear. So there are cells in superior olive which respond to the time delay, what is called the binaural delay. Okay. So one more cue that tells you the sound direction is the relative sound intensity. Suppose sound is coming from your far right, it will go through the right ear and without any loss of energy. But for it to go through the left ear, it has to go around your head, which it can by diffraction. But it will also go through the head, but in the process it loses energy. The intensity of sound that you pick up right, from your on your left ear will be lesser than the intensity that you pick up in right ear when the sound is coming from the right, from your right side. So based on that relative sound intensity, also you can find out where the sound is coming from, and some of these functions occur in superior olive. So when you go to auditory cortex, so just like the even at level of basilar membrane inside the cochlea, so you perform some kind of a Fourier transform the input signals. Even in the auditory cortex, there is a kind of a similar Fourier transform like representation is observed in the auditory primary auditory cortex. So you see that different frequencies are mapped onto different parts of the auditory cortex in almost a linear fashion. And so such a mapping of frequencies along the length of the primary auditory cortex is called tonotopy. The property is called tonotopy, and such maps are called tonotopic maps. Just as you have these orientation maps and ocular dominance maps and all that in the visual system, in the primary auditory cortex, in the primary visual system, that was in primary visual system. In the primary auditory cortex, also you have tonotopic map. So in addition to just in these neurons in this map, they respond to frequencies, single frequencies, right? So for example, neurons here, let's say here, they respond to 500 hertz, and neurons here respond to 16,000 hertz. But in addition to the frequency, they also respond to more complex features, like for example, modulations of amplitude and frequency and so on. So now people, just like people have looked at CNNs and their performance on image pattern recognition and compared with the visual system, right, in humans and macaques. So here also they looked at performance of human performance on word recognition. This is spoken word recognition, and again have done similar comparison. And again, they see that similar observations can be made by this comparison. So in the plot on the left side, you see the humans' proportion words correct. So many of the words are recognized perfectly when humans heard them. And that's x-axis, and y-axis is the number of words recognized correctly by the network. And you see that there is a very high correlation. So this gray line is perfect correlation, but what is found is actually something like this. Okay, and then if you look at the, so there is a, yeah, so here what they do is, they look at two categories of problems. One is word classification, and another is genre classification. So just like in the previous case, they looked at object classification and image classification. So image is more restrictive and object is more general as a broader class. So here also they take the word genre and the specific word and how to classify that. So for that, what they found is if you want to construct an optimal network to which will give high performance, instead of having totally different networks for word classification and genre classification, they found that it is good to have a shared layers between the two networks up to some point. After that, you split it into two separate pipelines or separate pathways, one for word classification, one for genre classification. And you can play around with it, play different, with different architectures and find out, which is an optimal architecture. What is the right point at which you want to split this hierarchy and have separate pathways for words and for genres. So again, they have done the same kind of exercise. So they looked at the shared, so they looked at the different layers, the response of different layers in the network when they train. And so then looked at different areas in the, from fMRI, right, from the human auditory cortex. And they found which ones give you highest correlation. So the shared layers give pretty decent correlation, right? And that is some of these layers. But beyond a point, so these are the, so you have the word branch here. This is a word branch, and this is the genre branch. So word branch and genre branch both give good correlation up to this point. After that, this the correlation drops down. Okay, so again, so look at the left part is about visual pattern recognition. So that's a different problem. Let us leave it now because we already discussed visual performance. Let us look at auditory domain now. So if you look at the correlation with spectrum, how well can you predict the spectrum, the frequencies present, the peak frequencies present in the word sound, and see which layers can estimate that best. They found that the lower layers estimate the spectrum best, right? But as you go to higher layers, they're not able to estimate the spectrum. That's obviously because as you go to higher layers, they're representing something that is much more abstract than the spectrum. It has got rid of the spectral information. It is focusing more on the word content, right? So therefore, you have poor correlation between activities of the higher layers and the spectrum. So similarly, for spectrotemporal modulation, changes in spectrum. So there again, hidden layers, middle layers, right, have higher correlation, are more able to predict that than early layers and late layers. But if you look at word recognition, right, the word branch has done better, right, than the genre branch. Now they've also taken kind of a word independent or a task independent kind of a correlation study. So the network is trying to recognize words and single words or word genres. It was not trying to recognize the speaker. But if you take these things, the activities of different layers, right, and try to correlate with the speaker to see or predict the speaker identity, they were able to do best with the again higher layers, right, and not as much with the lower layers. So that means even speaker identity information is more present in the higher layers of the network. Okay, so that's the kind of thing you can do even with a simple deep network. So which is a very potential tool, and people have done similar studies even in case of hippocampus and so interrhinal cortex and perirhinal cortex and all that. These are cortical areas close to hippocampus, and here also people are able to get very good results. So it's not just a sensory function; you can also go beyond that. Uh, so to summarize, backprop is widely used by DNN. Backprop algorithm itself may not be realistic, but the kind of networks you get as a result of backprop, they are pretty good. They are very valuable in understanding what kind of representations the brain generates. So yeah, so I think that's a very interesting paper. I can share the PDF, right? You can take a look at it later. So from tomorrow, we'll start a new topic. We'll look at Hopfield network and how it can be extended to understand the function of hippocampus, which is a major memory-related structure in the brain. Thank you. Now, I think Sandeep and Sundari will discuss the mid-sem questions.